Hello there, Dr. Dan Guerra from Med from the beautiful Pacific Northwest of the United States. Today, we're going to jump into another story about autophagy. We've been following along on this trend. I think this might be the third in a row, where basically we've been talking about how cells destined toward a certain fate. Today, we're going to emphasize how lipids play, big surprise, a very significant role in this destination. So we're going to describe some background information about why this is significant in relative to autophagy. Then we're going to talk about the disease, which is going to be front and center for this talk, which is, of course, a neurodegenerative disease we all are quite aware of and familiar with, Alzheimer's disease. And then we're going to go back and retrieve from our memory vault the discussion of sphingolipids, particularly ceramide and dihydroceramide. That goes back about 4 lectures. So those are the 3 main elements to put together for this talk. So why don't we just get started with it rather than saying we're going to do it. All right, that's me, Dr. Dan Guerra. That's the background there where that picture is taken is off my back porch, and that is the land leading to the Clearwater River, where we live, right on the edge of. This is Med, and this comes from Med Studios exclusively to you for free. And what I do is I provide these video lectures on YouTube so that it will stimulate interest in our company, because what this should showcase for you is how we are capable of going into the scientific literature and extracting from it very recent published research on any given topic that you may be interested in or that you need to research. So this goes to graduate students, postdocs, as well as medical students, as well as residents, as well as physicians themselves that want to know more about science behind a given therapy, be it pharmacotherapy, via surgery. And of course, for our larger audience of regular lay people, if you want to know more about biomedicine in general, or you have a specific question in mind that you'd like answered, all you need to do is contact us, and you'll directly talk to me. I'm a PhD biochemist with over 30 years experience as a researcher and as a professor. And myself, along with my colleague, who is a registered pharmacist, will be able to deal with your questions and hopefully dig into the literature sufficiently so that we can answer them with full vetting power. So that's what we do. That's our logo. That's our website there, www.veramed.com. That is the official email that you'll get when you go to our website. You can use that to contact us, or you can contact me on my email directly, which is down here. It's a Gmail account. I usually answer it within 24 hours if you go that route, and I'll be glad to answer any questions to get you started on your road towards using Med. We're a very unique service, and we think that we provide top-notch, in-depth understanding of what's going on in research science, particularly the natural sciences, particularly those associated with biomedicine, biochemistry, cell biology, cell physiology, pathophysiology, and disease. To name only but a few, actually. Major ones. Today's the 18th of February. As the date shows up there, that's actually today's date. It's late afternoon here. And this is my Facebook page. If you want to go to any of my previous Med lectures, you're going to find them there posted. I also have a Twitter account where they're posted. And I'm in the process of linking these onto our webpage. But since they're on Facebook, that's probably the easiest way to get to them. It's a public domain, and you can get these for free. But please contact me if you have any questions about them. I'd be glad to answer them. So what are we going to talk about today? Well, there's the heading, Lipid-Mediated Autophagic Post-Destination as a therapeutic mechanism to dissociate aggregating proteinopathies in Alzheimer's disease. So proteinopathies, that's just kind of a newly minted term in biochemistry. And all that really means is some pathology of proteins in a cell. It covers a lot of ground. It could be quite variable on the discrete and specific nature of that pathology, protein pathology. So if you haven't heard that term before, don't worry about it. This, of course, is another one of our shots off the top of wonderful Pikes Peak, where we used to live down there in Colorado Springs. Visited it recently. Beautiful place to go. All right. So we're going to talk about neurodegeneration and determine whether or not if it can be diverted via a reprogramming of cell fate. It's basically what we're talking about here. It's taking an autophagic destiny and using that to be involved as a therapy for controlling neurodegenerative disease. That's the topic of today. Of course, altering cell fate, as we talked about in previous lectures, can also be used to stop and block cancer and also invasion of microorganisms. So this is just one of the possible biomedical pathophysiological responses. So today it's neurodegeneration. Tomorrow it could be prostate cancer. All right. This is symbolic of what we're going to do. We're going to reprogram, just like this used to be white spruce and Douglas fir and red fir, trees that I cut down in the forest, in the Clearwater National Forest, not far from here. Cut those logs up into blocks and then split them for firewood. So that's now the firewood, those building blocks there, just like an autophage, it can be used for something really important. In our case, rather than for bioenergetics, it's for heat of the house. And that's how we heat our house here. All right. So the paper we're going to talk about is actually not due in the library, in the electronic library, until March of 2018. But as you probably are aware of, some journals go ahead and pre-publish their papers online, and this is one of them. I believe this is, however, under a paywall. You have to have a subscription or have another access to this to be able to get pharmaceutical research papers. So this is going to come out in March of 2018 in paper form. And there is the citation there. So let's take a look at the general introduction before we get ready. So we all know that Alzheimer's disease is the most common age-related neurodegenerative disease with associated incremental or hierarchical dementia. At the protein level, AD, which I'm going to call Alzheimer's from now on, is linked to both neurofibrillary tangles and beta amyloid plaques or deposits. Although, even though it's associated with those two, a direct causal mechanistic link to these proteinopathies, these are aggregated proteins, which result in neurofibrillary tangles and beta amyloid plaques. Those are proteinopathies. It's not universal. So there are people that show signs of Alzheimer's disease, or AD, that don't necessarily have one or both of those proteinopathies. And then there are people that have those proteinopathies that don't show outward measurable neuropsychiatric signs of Alzheimer's disease. And we know this from autopsy. So it's very interesting, right? But it doesn't mean that these two protein aggregation issues are not very important in AD. And in fact, they probably, if not, are not causal. They're very tightly correlated. And then tonight we're going to find out a little bit why. So the majority of AD, that's 90-95%, is idiopathic. We don't know where it comes from. It's random in terms of who it attacks. And the real major contributory factor is if you're over 65. So you need to be over 65, and every 5 years after that, the potential for you to get AD increases, about doubles. But even in the end, only about half of human beings seem to get AD if they live that long. Okay, so people can live well into their 80s or even into their 90s and not have AD. For example, my mother's 92. She shows no signs of it at all. My father lived to be about that age, and neither did he have any AD. So it certainly isn't universal. It does seem to run in families, but it nevertheless is idiopathic. We don't know where it comes from. There is a rare congenital form, I should mention. It's called familial Alzheimer's disease, or FAD, and it does have presumed genetic loci. So that is more tractable to measuring disease spread and disease ontology, because we know more about the genetic loci. We know a lot about the genetic loci of the idiopathic common form too. It's just that we can't really say with absolute certainty. Of course, if you know how I feel about certainty, nothing is absolutely certain. Everything is absolutely probably uncertain, except for that statement, which is certain, although that's uncertain. At any rate, we're not going to talk about FAD. We're going to talk about regular AD. But all AD is characterized with age-related progression of protein aggregate accumulation. So it has a family recognition with other neurodegenerative diseases because they show very similar things. Parkinson's disease, PD, Huntington's chorea are two other very common, although the Huntington's chorea are far more rare than AD, proteinopathies that result in from neurodegeneration. Anyways, those proteinopathies obviously implicated in pathoproteostasis. What do I mean by that? Pathoproteostasis means you have a certain homeostatic level of proteins, both in concentration and in variety in a given cell type, like say in a glial cell or in a neuron. And that static control of those proteins, what happens is that breaks down. When that breaks down, that's when you get a proteinopathy. And often it involves aggregation. So pathoproteostasis can occur because of a couple of different mechanisms. Corrupted protein turnover, as with the ubiquitin proteasome system. So proteins will turn over as well as they should. Excessive protein synthesis, for example, a constitutive mTOR pathway, which has been recognized and shown, or with incompetent autophagy. And that's going to really be the key highlight of today's talk. We've talked a lot about autophagy, and because of that, I'm only going to briefly go over it, although I think I will be able to go over it so you don't need to go back and look at previous lectures. But certainly there are plenty of places on the web where you can look up what autophagy is. But if you want to see, for example, how I've been covering it, go to one of my previous video lectures shown there on that hyperlink. All right, on with the classes. All right, so this is a nice cartoon showing you the transition towards Alzheimer's disease, or AD. And it's from the point of view, somewhat myopically, of the A beta protein. So that's a really important thing to understand. This is an amyloid beta peptide that seems to accumulate and aggregate. It seems to be linked to your general mainframe AD. Okay? So some kind of oxidative stress, which just happens in the presence of molecular oxygen, it's always there, right? 21% of the air you breathe, results in redox-sensitive transcription factor control, that is, expression at the RNA level of the APP, okay? So that's the amyloid precursor protein. Now, in order for it to get processed to the A beta, which is considered the toxic form, and its aggregation products, or toxic form, it has to be processed by an enzyme called secretase. It's called secretase because it has to be secreted and packaged along with the amyloid precursor protein. So you package secretase, the enzyme, which is a protease, with the APP. You proteolytically degrade it to make that beta amyloid peptide. And when you make that, then aggregation starts, and then you see frank disease. That's the idea there, okay? So you get transcription of the APP, which is a precursor protein. It gets fresh transcription caused directly by reactive oxygen species and some other pathways that involve protein kinases and elongation factors and protein synthesis to turn on the expression of the secretase, called here base 1. Those two combined then make the A beta 42, which is your canonical A beta protein, which is going to be involved in this disease. Now, that just the presence of that protein can trigger autooxidation events using these metals, copper and iron, which can, of course, exist in multiple oxidation states in the cell, and which are found in the cell, usually as heme-containing proteins, right? Not as free forms. They, of course, will contribute to oxidative stress, thus increasing the unbalance and increasing the amount of A beta, thus causing a vicious cycle. Now, that A beta protein can also activate microglia. Remember, those are basically resident macrophages in the central nervous system in the brain. When that happens, microglia can further cause oxidative stress because they are what? They're macrophages. And they work with oxidative bursts, the production of pro-inflammatory cytokines, just like any good macrophage will. They also, A beta protein, will also directly impact mitochondria. It'll decrease or tank bioenergetics, and at the same time, because of that, it'll uncouple electron transport chain from ATP synthesis or phosphorylation, oxidative phosphorylation, and because of that, you build up partially reduced forms of oxygen known as ROS, reactive oxygen species, which further causes the oxidative stress, which further causes this super imbalance, making a whole lot of this A beta from the precursor and from this enzyme, which forms the A beta toxic form. Now, when things are going well, the A beta protein can be cleared. Okay? And when it's cleared, then it's out of this whole terrible raging pathway. And when it is removed, that's when you start to see less of a AD disease, right? Now, very seldom, if ever, do you see remittance of AD. So when I say that we get clearance, it means for people who suffer from AD where the disease doesn't progress very rapidly. I really can't think of any place in the literature that's shown that we get actually remission of AD once it starts. At least nothing I've seen in the literature, and I have looked for it. So oxidative stress associated with increased expression of the precursor protein with the secretase leads to the accumulation of A beta 42. Clearance of it from the brain is also blocked by oxidative stress. So remember that even to clear it, the whole oxidative stress process blocks that. That has to do with the whole machinery involved, such as the proteasomal activity. Anyways, association of the A beta 42 with mitochondria, microglia, and metal ions, as I've been saying, leads to further metabolic stress and ultimately oxidative stress feeding this beast. All right, now this slide came from one of my previous talks when we talked about autophagy. There's the reference, or there's the link to be able to get this figure for yourself. Remember that autophagy is this process down here. So rather than having growth stimulation via growth factors or via hormones like insulin, where you're turning on the mTOR pathway and you're anabolic, that's shut down because you have a nutrient depletion or some kind of stress. It's shut down because of the activation of adenylyl monophosphate kinase, which shuts down the mTOR pathway. Because of that, mTOR can't block what's going on down here, which is the aggregation of the ULK complex, picking up an autophagy protein called 9L, and then causing then the second complex in the autophagy process to be formed, this one here with the Becklin, the ATG. We talked about this again at some length in the last talk. This complex, in combination with the ULK complex, then generates the phagophore. That's that membrane fragment that can come from the ER, the Golgi, even the mitochondria, and causes a biophysical alteration of that membrane complex so that it binds, remember, to the phosphatidylethanol or the phosphatidylenacetyl phosphates in that membrane fraction, start causing a curvature of that membrane by binding all these proteins covalently. Okay, so this is called the lipid complex. Once it does that, the membrane starts to fold upon itself. It's a biophysical process. It's a protein-lipid interaction. It starts engulfing or trapping cytosol. When it does so, it can entrap entire organelles, like shown here, mitochondria or polyribosomes. Ultimately, the autophagosome closes with the membrane. When that happens, more interactions with protein-lipid happen, and the lysosome is then taken into the autophagosome to make the autophagolysosome. Remember, the lysosome has all the degradative enzymes. It's going to destroy all this macromolecular structure, making all those monomers, like my wood splits on that picture I showed you a few minutes ago. Now you're ready to use all of these building blocks like amino acids, like fatty acids, like glycerol, like phosphatidylcholine, like palmitoyl-CoA and serine, to be able to rebuild proteins and lipids, and in fact using the nucleotides, the purines and pyrimidines to resynthesize RNA and DNA. That's how degradation occurs, and that's what autophagy is all about. It's a limited destruction or self-eating of the cytoplasm or just of the organelles themselves, but a maintenance of cell survival this way, not programmed cell death. So autophagy has been linked to all these different kinds of potential biomedical concerns and phenomena, as well as normal physiology. But autophagy can, though, lead to cell death directly. It can ameliorate cancer, or it can lead down the cancer deadly pathway. It feeds into the innate immune system because of the degradation of all of these macromolecular structures can turn on the innate immune system. It's been associated with cardiomyopathy, such as hypertrophy, cardiac hypertrophy. Autophagy, of course, is linked to aging, the control of aging, but also the extent of aging. Autophagy also can help remedy infectious diseases by hooking up with the immune system to digest invading microorganisms, or in fact, sometimes it overreaches its mark. Autophagy is also involved in metabolic diseases like type 2 diabetes and non-alcoholic fatty liver disease, and in the neurodegenerative diseases, as we're going to talk about today. So multiple strategies for autophagy to control, and during that control process, actually to promote a more healthy state, okay? Or sometimes it can proceed onward and actually make it for a pathological state. So now let's link autophagy with AD proteinopathy. Dysfunction in autophagy results in poor clearance of the harmful amyloid beta or A beta aggregate. Indeed, neuronal A beta contingent decreases with increasing autophagy. Okay, so they are bilaterally opposed, right? So the neuronal AB contingent, right, the pool of AB decreases the more autophagy you get, because what does autophagy do? It breaks down the aggregated protein, which is what you want when you want to inhibit neurodegeneration. In general, dysfunctional autophagy is observed in human central neurodegeneration. So if you have dysfunctional autophagy, that's a problem. That's why we're talking about it today. Now, the functional autophagy tracks pathogenesis of AD, where it serves to remit, okay, so it can remit the proteinopathies. It can remit A beta, and tau is another protein that aggregates. That's in the neurofibrillary tangles, and neuropsychiatric outcomes such as improved learning and memory. So if autophagy tracks the pathogenesis and pulls it down, pulls it back, AD never progresses to that outward sign of severe limitations in learning and, of course, eventually loss of memory. So we know that there is a link there, and it seems to be linked directly with removing these aggregating proteins, the A beta and the tau proteins. Okay. So there are three types of autophagy, and I want you to know them. There's a chaperone-mediated, or CMA, shown here, where a protein acting as a chaperone helps chaperone proteins into the lysosome. Okay, so as it says here, it degrades specific, not specific, cytosolic proteins containing the CMA motif. So if the protein has a certain amino acid signature or functional domain, functional structural domain, it can be picked up by that chaperone, loaded up with the lysosome, and then that ultimately connects with the autophagosome, and you get degradation, okay? So that's one way you go through the process. LAMP2A translocation complex actually is necessary. That's one of the proteins necessary on the outskirts membrane of that lysosome organelle. Then you have microautophagy. That's something I talked about a lot in the last talk I gave, and that's basically getting an autophagy of peroxisomes, mitochondria, endoplasmic reticulum, polyribosomes, okay? Those also feed into the complex. So these can all be happening simultaneously, or they can be acting autonomously, okay? Then there's general macroautophagy, which, again, hopefully you understand by now. We just looked at it. All these proteins have to be linked in order for that phagophore to come around and make the autophagosome, and eventually link up with the lysosome and make an autophagolysosome and complete the circuit, complete the process of autophagy, thus decreasing the opportunity for aggregated proteins to cause disease, okay? So macroautophagy eliminates intracellular components, proteins, organelles, lipids, by both selective and non-selective mechanisms. The substrates are enclosed in double membrane vesicle we call the autophagosome. Once the substrates are engulfed inside that, they fuse with the lysosome, and then you're finished with the process. Now note, alterations to autophagy pathways have been described for several neurodegenerative diseases. Okay, this is really important. When I say alterations, I really mean dysfunction. And indeed, these defects in the autophagy machinery can occur at different steps along the pathway anywhere. Now you see here, when you look at the fine tuning of this, I got this cartoon from a paper published in Seminars in Cell and Developmental Biology in April of 2015. Okay, so that's where I got this slide from, or this figure. Notice that AD, Huntington's disease, Parkinson's disease, along in association with those proteins, are things that block this autophagy pathway. Parkinson's disease blocks this selective cargo mitochondrial aggregates, ER. This is the micro autophagy we talked about the last talk I gave, that I gave you the hyperlink to. Here, once again, Huntington's disease, motor proteins are somehow blocking this component of the process. Same disease, along with AD, in the presence of the protein RAB5, blocks this fusion with the lysosome to the autophagosome. So see, there's multiple places where these disease states, you see, these are disease states, along with their accompanying proteins. There's ApoE4. It's another important, that's an apolipoprotein that works early in the pathway of lysosomal organization, leading to autophagy. And anytime that these diseases are rampant, these multiple places along this pathway can be blocked. And that's something we know well, and it's encompassed in this paper when you want to read it. Now, we have one more component to add, sphingolipid metabolism. Dihydroceramide and ceramide, ceramide 1-phosphate, sphingosine 1-phosphate, as well as all the gangliosides or glucocerebrosides, are all potent signaling lipid molecules that contribute to the pathogenesis of many inflammatory diseases. We've talked about these in various talks in the past. Now, dihydroceramide is a precursor to ceramides, the direct substrate to make ceramide. And that's in the de novo sphingolipid pathway. And the enzyme that converts hydroceramide to ceramide is dihydroceramide desaturase. And it's nicknamed DES, D-E-S, 1 or 2. There's 2 isoforms of them. They have some substrate specificity according to the fatty acid that is N-linked in those sphingolipids. Sure. So DES1 is associated with electron transport chain, and it includes the NAD and NADPH cytochrome B5 reductase and cytochrome B5. It's active at the cytosolic base of the ER as well, so that's where that enzyme resides. DES1 is, when you silence it, cells exhibit high levels of autophagy, in part due to impaired ATP synthesis, so that's the signaling, right? Because remember the AMP kinase. So when you decrease ATP synthesis, you use up all the ATP, you turn ATP into ADP into AMP, boom, AMP kinase comes on. And as I said, that leads to the activation that shuts down the mTOR and it turns on autophagy. That's right. Now, another observation to throw in here, the polyphenolic compound known as resveratrol, okay, very important new kid on the block as it comes to therapy for multiple kinds of disease states, including cardiovascular and cancer. Now here we're looking at a potential for it to be involved in neurodegenerative diseases. Anyways, resveratrol, which is a polyphenolic compound from plants, or indeed another component from plants, usually found in nuts, a vitamin E derivative known as tocotrienols. They may affect amyloidosis in the animal models. So that's another observation. We know something about those lipids, and of course we know a lot more about the ceramides. But they use resveratrol in this paper, so you got to know a little bit about it. It can actually induce autophagy after increase in dihydroceramides, where it may inhibit dihydroceramide desaturase. If you inhibit that enzyme, you're going to build up the dihydro form and not the ceramide form. Why is that important? Because ceramide causes cell fate to go towards apoptosis. And apoptosis is a bad thing in the brain, right? Because that's neurodegeneration. That's the death of the neurons. Don't want that. You want autophagy if you've got stress or damage, right? You don't want cell death. Sometimes you do want cell death, like in a cancer or in cardiac hypertrophy. You don't want that in neurodegeneration. So you want a different endpoint. So you look at autophagy from a totally different perspective. You want autophagy in the diseases of the brain. Okay. You do not want apoptosis. So we're going to see a dihydroceramide desaturase inhibitor called XM462. There's another analog of this called 461, which we're going to see. Just to recap how you make sphingolipids, you start off with plain old dreary palmitoyl-CoA coming directly off fatty acid synthesis or from the diet, for example. It reacts with L-serine. Okay, you make 3-keto dihydrosphingosine, shown here, and derives the reaction as carbon dioxide release, and CoA is gone. Okay, so now that you've got this keto dihydrosphingosine, you reduce it with NADPH, and you make sphinganine. Okay, you've now reduced this carbonyl here. You've got sphinganine. Notice you still retain this amino group here. It's very important because you're going to get an amide-linked fatty acid there. All right, here's the pathway. Palmitoyl-CoA serine. There's 3-keto-sphinganine. There's the NADPH doing the reduction to sphinganine. There's the really important ceramide synthase adding an acyl-CoA. That fatty acid can be anywhere from stearate to oleate to behinic acid to arachidonic acid, even to icosapentaenoic and docosahexaenoic acid. All different possible isoforms of the dihydroceramide. There's going to be the key molecule in today's talk. That is going to be promoting autophagy, that compound there. Now, what can happen to dihydroceramide is going to be desaturated. And we talked about this again in at least 3 other talks here at VeraMed. You're putting a trans double bond at the 4 position there. What used to be palmitic acid is now basically palmitoleic acid because it has a double bond, sure. But it's a trans double bond. And I told you that should help you understand that trans double bonds in fatty acids are not indeed toxic. It's a big mistake, and it's not true. The literature doesn't really show that if you look at it carefully. Anyways, there's a naturally occurring trans fatty acid you find in sphingolipids. That's ceramide, okay? So you got this added fatty acid, you've got this sphinganine backbone, and you've got this trans double bond. You got ceramide, very hydrophobic compound. Now that goes on to make all the complex sphingolipids, all the glycosyl sphingolipids, such as cerebrosides, galactocerebrosides, and glucocerebrosides that you find, for example, covering the myelin sheath. Right, all those really important membrane lipid components that are necessary for correct saltation in neural transmission, such as the evocation and revocation of action potentials. And sphingomyelin itself, of course, around the acting as that sheath molecule. All right, so this is a precursor to it. Now, there is some feedback where you can run back into this cycle when you break down these lipids. I'll show you that in a moment. There's an enzyme called ceraminidase, which now remove that fatty acid. Now you add a kinase, you got sphingosine 1-phosphate, and it has its own very intriguing nested set of potent biological properties as a signaling molecule. We're not going to talk about that today. We passed on it and landed on it hard in a couple of previous talks. So go back and look at those if you're interested. This is the final degradation pathway to this hexadecinal, this aldehyde, double bond aldehyde lipid, and then that's on the way out. No more biological activity. All right. So another way of looking at it, only now it's showing you that you can make ceramide multiple ways. There's a de novo pathway we just looked at. There's that really important desaturase enzyme, DES. But sphingomyelin can be broken down by, for example, acid sphingomyelinase. And when it does that, it will synthesize ceramide and phosphatidylcholine usually. You can also make ceramide from this pathway here, sphingosine 1-phosphate lyase, taking ethanolamine 1-phosphate and palmitolaldehyde, making a sphingotine 1-phosphate, phosphorylating that, okay? Well, actually removing that phosphate to make sphingosine, and then building back up the ceramide by picking up the fatty acid. Okay? So that enzyme is called ceraminidase when you break it down. We already saw that, but you can also rebuild from this. So just blocking DES, which is this reaction right here, doesn't mean that ceramide can't build up. That's a really important thing to note in this paper. Okay, and these are just compounds which are known to block these various enzymes. Okay. So one more of these cartoons and we'll get into the paper again. So again, there are multiple ways to synthesize ceramide. Sphingomyelinase or ceramide synthase, shown here. Ceramide is a precursor to the glucocerebrosides. You can make sphingosine 1-phosphate. If you make ceramide, though, the key feature, this comes from a 2008 paper in FASEB Journal, apoptosis. So ceramide is an apoptotic signaling molecule, very potent in generating programmed cell death. So let's get back to the farm research paper. It examines the effect of inhibiting DES1 in primary neurons from a double transgenic mouse model expressing human mutations in amyloid precursor protein and presenilin 1. Now, they use a transgenic mouse model because they want to have it overproduce those two proteins which have mutations in them so they'll make better aggregates. So that is the model for AD in the mouse. One very potent model. The neurons of these transgenic mice accumulate A-beta, see, as diffuse neuritic plaques in an age-dependent manner, and they display some dysfunctional neuronal proteins. It's all important to show that you've got frank disease. DES1 inhibitors, XM461 and 462, induce autophagy in both wild-type and transgenic neurons. Okay, so that's interesting. You block that desaturase, you induce autophagy. The induced autophagy is associated with changes in neuronal lipid profiles. For example, a reduction in ceramide, sphingomyelin, glucosylceramide, with a concomitant increase in all the dihydro forms of those. Okay, dihydroceramide, dihydrosphingomyelin, etc. Right? Okay. These changes in lipids track with increasing autophagic markers. So the more you make the dihydro forms of those lipids, the more you increase autophagy, and you see the markers go up. Consequently, there is a significant reduction of amyloid secretion in the neurons isolated from transgenic APP-PS1 mice. Okay, those are the mice that have been built in these transgenic model. First set of data. This is showing you that. Okay, first of all, you're looking at wild type shown here, and this is the transgenic. You're looking at resveratrol or the two DES inhibitors, and CO here is control, and you're looking at the relative activity of the desaturase enzyme. The control is here. Resveratrol shows a statistical decrease in it, as do the two desaturase-specific inhibitors. Now, that's in the transgenic line. We have an overexpression of, right, those A-beta proteins in the presenilin. Now, with wild type, the same trend. Okay, so DES inhibitors indeed inhibit DES in a time-dependent manner in both wild type and transgenic. Here's time-dependent. The longer you go out, the deeper you decrease the amount of DES1 activity. You want to get that decrease in DES activity because you want to build up the hydroforms. The hydroforms, remember, are faithfully protective. Now, this is nothing more than data from Western blot, Kumasi and MTT assays. Neurons from transgenic or wild type litter mates were treated with either resveratrol or the two DES inhibitors. After plating, DMSO is used as a control. For 24 and 48 hours, cell viability is determined from two methods, using Kumasi, which is just looking at protein, and this MTT, which basically is looking at the amount of metabolic activity of reducing equivalents in the form of NADH or NADPH. All right. Now, wild-type neurons were not affected by any compound for 24 or 48 hours. Okay. So you don't see any effect on the wild type. Glasses back on. All right. Shown here. Okay. So there's your control. There's the percentage subject. This is cell viability. This is the Kumasi staining. No effect. Now, and even with the transgenic line, you do see a little bit of a decrease using resveratrol or the desaturase inhibitor, but not so bad. But at 48 hours, you do definitely see a reduction in cell viability using the Kumasi staining, and this is more pronounced using the cytotoxicity assay, or the MTT. So, a more pronounced effect was observed on transgenic neurons for all the treatments after 48 hours. Okay, so that's out here, right? With resveratrol and the two desaturase inhibitors, each provoking a significant reduction in protein, reaching 12, 24, and 20% respectively, okay, of that entire system. Now, cytotoxicity was evident using the MTT assay, which measures the metabolic activity of NAD and NADPH-dependent cellular oxidative reductase enzymes, and that's mostly basically looking at mitochondrial bioactivity. XM461 and 62 both proved to be negatively altering mitochondrial metabolism. which is what you would guess, okay? That's because autophagy is being kicked in, okay? Because you're jacking up those dihydro forms of ceramide, and that's going to induce autophagy, and that's going to be resulting in a decrease in oxidative phosphorylation. Okay? Even after 24 hours, with XM462 proving to be the most toxic, results were similar in wild type and transgenic neurons after the 24 to 40 hour marker. So there's some variation here, but basically you can see that this 462 is more cytotoxic than 461, and both of them are more cytotoxic than the resveratrol alone. Here's your Western blot. Some reduction in the APP in wild type, and this was enhanced after time course, especially with 462. That's the inhibitor. So you look here, you add that inhibitor, you're going to get a real decrease in APP. That's good, right? Because that means that autophagy is working. So you add the desaturase enzyme, you get more of the autophagy coming up. Autophagy is going to remove the proteinopathy, that is the aggregated APP. This P120, synaptophysin, beta-synapsin, those are all proteins involved in in the synapse, right? And so you want to know whether or not those proteins are being degraded. In the wild type, you see here that when you add this drug, you get a decrease in that. And with the transgenics, the same kind of thing happens, okay? With some of these really important synaptic proteins. So synaptic proteins are similar to the profile that we've seen with the amyloid precursor protein. And the bottom line is that wild type showed a major drop in synaptic proteins with these inhibitors. So that's one of the problems with it. You add these inhibitors, right, you're going to get a decrease in synaptic proteins. Why is that? Because you're getting an increase in autophagy. What the autophagy, though, is hopefully doing is getting rid of the more toxic aggregated proteins. These are not toxic at this stage, okay? But just showing you that you will get a decrease if you add a desaturase inhibitor, okay? That's what basically it's just showing here. There's P120, you see a pretty good reduction. Synaptophysin, pretty darn good reduction compared to the control, and synapsin the same way. All right. Now, the decreases in ceramide with resveratrol and with the inhibitor 461 were concomitant with increases in dihydroceramide. So what you see here is that when you add the inhibitor, right, you get an increase. Okay, here's resveratrol. Here's 461, right? And then you look at the transgenics versus the wild type. The wild type, here's the control. You add resveratrol, you get a lot less of ceramide. Okay, so that, again, it's blocking the production of ceramide. And you add the desaturase, it blocks it, but not as much as the resveratrol. The transgenic line, even more severe features occur. All right, that's just the C18 ceramide. That's total ceramide, same basic trend. This is now the substrate for that reaction, or the precursor, and what you want to see is an increase in it when you're adding resveratrol or the desaturase inhibitor, because this is the substrate for the reaction. You do see increases both at the C18 level and at total ceramide level. Look at sphingomyelin, same kind of basic thing. Bottom line is that the DES inhibitor was rather consistently knocking down products of the inhibitor reaction and concomitant jacking up substrates as per mode of action. So it's blocking, remember, the desaturases, and so it's going to block the production of sphingomyelin because that has that trans double bond at the 4 position in the sphingosine backbone. The C18 dihydrosphingomyelin is going to go up because, remember, this is the precursor. So because you're blocking the enzyme, you're going to see higher levels. You can see a really whopping increase here from using the desaturase inhibitor. And these are glycosylcerebrosides, again, show with that same, they have that same desaturated product, that delta 4 trans double bond. And indeed, you do see an increase in the transgenic line and even in the wild type by adding the inhibitor or by adding resveratrol. All right. So now we're looking at analysis of the autophagic markers themselves. So you have neurons from transgenic or wild type littermates treated with resveratrol or the 2 desaturase inhibitors, and DMSO was a control. After 24 to 48 hours, the cell extract is obtained and analyzed. Again, these are Western blots. using antibodies against LC3 and P62. These are important enzymes involved in autophagy. Okay, and SPS6 is a reporter of S6K activity. Of course, that's involved in the whole mTOR pathway. And beta-actin gap dehydrogenase, because the other 3-phosphatidehydrogenase are just used as loading controls. So here's the wild type. Data suggests that the XM461, okay, shown here, and 462, shown here, okay, enhanced autophagy. See, you're getting rid of the piece, the product of this phosphorylated S6. You're getting rid of it when you add either resveratrol or the 2-desaturase inhibitors in the wild type. See the same thing in the transgenic line. In 24 hours, 48 hours, you completely lose, really, PS6. You reduce P62. These two proteins involved in the PHAGA4 metabolic pathway, again, you see decreases, and this is shown in these histograms, basically. So when you add resveratrol or the two inhibitors, and you compare transgenic versus wild type cell lineages, you get a decrease. Every time you get a decrease, and it looks to be time dependent in the overall, in all these proteins that you would expect to be taken out, right? Because these are autophagic markers. And so you're seeing autophagy is working well by inhibiting the desaturase enzyme. So using an inhibitor of autophagic flux, which blocks lysosomal activity specifically, the amount of LC2 and P62 immunoreactivity increased after a 2-hour exposure to bifidobacillin relative to the untreated controls. Okay, so the increase was even more evident in neurons treated with either of the 2-desaturase inhibitors. Both of those are efficient inducers of autophagy. In contrast, the resveratrol wild-type neurons had a low autophagic flux and basically a low degradation of P62. So it looks like resveratrol is now falling out of being a good pharmacotherapeutic for enhancing autophagy and annihilating those proteinopathy aggregates. Okay, now that was all obtained, that data I wrote here was shown in Western analysis. I didn't show you the data. All right. Now, another very important thing, how much DES activity you have, and is that linked to beta amyloid secreted? Okay, so after 24 hours, resveratrol doesn't seem to have any effect on the beta amyloid compared to the control. There is some decrease by the 2-desaturase enzymes, but look what happens at 48. Resveratrol and the 2-desaturase enzymes greatly reduce the amount of amyloid beta secretion. This is the end product of the bioassay for looking for Alzheimer's disease. So if you're getting these histogram lines dropping, these caving in by using these desaturases, even with the resveratrol, that's a good thing because that's the target. The target is to reduce the amount of amyloid beta. All right. Here's your dialectic. Inhibitors like XM462 can induce autophagy through dihydroceramide dependent and independent pathways. That was seen when you looked at the molecularity of all of those slides. Some things go up, some things go down. And you know why. Because you're learning lipid biochemistry. You don't make ceramide one way, just with the desaturase. You have 3 full pathways involved. Desaturase is only the linear de novo pathway, right? So you can make it, remember, from sphingomyelin. You can also make it from degradation products directly from the sphingosine metabolism pathway and from glucosylcerebrosides. So it's not the only way. Now, resveratrol showed only a tendency to enhance autophagy in wild-type neurons. And in contrast to the DES1 inhibitors, resveratrol did decrease ceramide levels but barely increased the reactant to that. Now, that's unusual. That's interesting. So it decreases ceramide, which seems to be good because you don't want apoptosis, but it didn't really do much with the precursor. That's because it's not specific. That's because resveratrol is not directly targeting the desaturase, whereas those drugs are. Because of multiple potential, and this is how I answer it. This is now the follow through. This is the payoff in the dialectic. Because of multiple potential metabolic pathways, for ceramide levels to change, they cannot verify this desaturase inhibition will consistently function in humans because of those three possible pathways from ceramide to and from it. And the resveratrol is far too broad in mode of action to make that faithful comparison. So now I'm going to remind you what the mode of action is, resveratrol. The mode of action of that polyphenol is linked to its effect on enzymes that control gene expression globally. The MOA is to activate a deacetylase, such as a sirtuin. Those are deacetylases in the cell, sirtuins. Hence, resveratrol will promote deacetylation of histone proteins that are associated with chromatin. So doing causes that DNA to become condensed, and it decreases expression of genes from that particular locus. If the induced transcription of DES is not decreased by resveratrol, but rather other competing pathways are blocked in expression, which could well happen because this is a global nucleosomal response, right? It's a chromatin activity. A relative increase in DES could occur. So that's why you get variation with that resveratrol. This may only partially explain the transgenic anomaly in the study, and is thus a flaw. It's a flaw in the study. It's also a flaw, I think, in the reasoning. I think they were thinking resveratrol is going to work the same way, and even the desaturases are going to have a very linear effect. And why would they? Because you have three pathways leading to and from ceramide. That's my point. Now, to back that up, a paper that just came out in Journal of Lipid Research, published just at the end of the month of January. Again, it was only 21 days ago exactly. And the title tells it all. Inhibition of acid sphingomyelinase disrupts lysosomal nutrient signaling complex, the LINUS, and triggers autophagy. So this paper basically supports the PharmSci paper we just went through, since a decrease in ceramide levels from an alternate pathway indeed enhances autophagy, presumably by blockading apoptosis. So again, if you inhibit the acid sphingomyelinase, you decrease the amount of ceramide. So that makes sense because that's what the enzyme does. Remember, ceramide is not going to give you autophagy, it's going to give you apoptosis. So you block apoptosis, and then the cell fate is then reprogrammed to go into autophagy. See? Cool. That I found that paper is even more cool. Here's a JBC paper from 2 years ago. So boy, is it ancient, right? Get it like a walker or crutches. Now that's not a very old paper. I'm being silly, right? SIRT3, which is one of those deacetylases, a sirtuin, mediated deacetylation of ceramide synthase, increased enzyme activity and ceramide accumulation. So if you remove an acetate from the enzyme ceramide synthase, you make the enzyme more active. So, whoa, what about that? Resveratrol actually activates deacetylation, which means it activates ceramide synthase. So this pathway would be enhanced if SIRT3 is specifically activated by resveratrol in the paper we just looked at. So this is one of the underlying features that they didn't cover in the PharmSci paper. We're covering it because I dug into the literature and found the road to the mechanism that leads to this apparent paradox. It's not a paradox at all. One more paper to bring up, it's in PharmaZ 2016 September, which I found, demonstrated that indeed SIRT3 is activated by resveratrol, so that ceramide synthase should be activated. Okay, that's the important point. However, this is how we get out of this. And why the paper, PharmSci paper, still worked with resveratrol. The ceramide synthase enzyme actually catalyzes the synthesis of not ceramide, because you need desaturase for that, right? How silly. The enzyme name is not, it's equivocal. And that equivocation is because you need desaturase to finally make ceramide. So actually what ceramide synthase does, it makes dihydroceramide from sphingolipid. Should be a P in there. And acyl-CoA. So this might explain the role of RSV in autophagy. See, so this paper, the JBC paper, and the PharmaZ paper clears up why the PharmSci paper looked paradoxical. Because, and it also helps clear up that previous paper we just looked at, because ceramide synthase doesn't really make ceramide, even though the name says that. It makes it dihydro. And we already just told you, we've been telling you again and again, the dihydro form is autophagy, not apoptosis. So you see, that's why you get all this breakdown of all of those aggregates. One last critical point. This is very important. This again comes directly from the hip. That means it's coming directly from my synthesis of this information. And also, I think it was nodded and it winked at a little bit in some of these papers. Sphingolipids play a role in lipid raft formation. And the rafts, of course, transport, at the very beginning of the process, the precursor protein, the amyloid precursor protein, and the secretase, so that the actual production of the A-beta-42 is directly linked to lipid composition of the raft. What lipid composition? Sphingolipids, you see? So the raft, the biophysical formation of the raft, is dependent on sphingolipid molecular species. Right? So if you go losing certain lipid species, you're going to lose the integrity of that raft. Therefore, you're going to lose the ability to synthesize the A-beta protein from the precursor, because you may not link up the APP, the amyloid precursor protein, with the beta secretase. So it's way up at the beginning. So I say, therefore, that the inhibition of DES1 or activation of ceramide synthase might reduce beta secretase activity or reduce the memory domains containing APP and beta secretase, impeding the amyloidogenic processing of the APP. So this is an added positive effect, right? When you start retailoring those sphingolipids, you might actually prevent the substrate or the platform to make the protein which will aggregate, right? The peptide, the A-beta. Furthermore, the pharmacotherapy might enhance the autophagic flux. This just logically follows, which leads to the generation of less A-beta peptide. Okay, basically, we get this now. DES1 might be a novel pharmacological target, so inhibitors like XM461, which look really potent in this paper, although it did cause some cell mortality, could be neuroproductive in AD by enhancing autophagy. So it looks like there's a whole lot of food for thought here about maybe targeting that ceramide pathway so that we can divert the pathway away from ceramide and directly into the dihydro forms. Doing so links it up to autophagy. Autophagy is going to be neuroprotective, and so maybe that could help in Alzheimer's disease. So see, ultimately, this has a clinical feature. All right, I'm done with the talk. That's me again. There's my email address, and there's my Facebook. There's the company's email address. Contact us either way. Website, remember, we're scientists verifying published evidence in biomedical sciences. That's what we do. Right? Of course, it's what we do. All right, so let's shut this whole thing here, make it small. And all I've got to say now is bye for now. Thank you.